There we go. All right. Good, good, good. All right. You notice that your exams have been scored. They've been posted. You should be able to get into them. I think you have. I think I set it up so you have a week to get into them and take a look at them. So please do that. As I said in class, I mean, I've been giving these exams. I have a host of exams, but I've been giving all of them for a number of years. I think I found all of the ambiguities or problems or mistakes. But if you find something you don't understand why I scored it the way I did, or, in the essay question, if you don't understand how I scored that, I basically grade your essay questions the same. Well, not basically. I use the same little rubric. There will be a rubric that'll pop up and it'll show you how I scored you. And you get points for, obviously, abiding by the length. And that's not just an anal thing where I'm sitting there trying to count words. Those are the amount of words, I sit down and I write these answers to myself, and I say, that's the amount of words I think you should use to adequately explain the concept. So if I say 200 words, I'm not looking for you to say the same thing over and over again to get up to 200. I'm saying, I would think to explain this, you need about 200 words. So that's a good way to judge depth, of detail and that sort of stuff. I give you points for grammar, punctuation. I'm not real stickler about that, but if it detracts from my ability to understand what you're saying, especially if it's a lot of fragmented sentences or just blurbs, that kind of stuff, then obviously that becomes a factor. And then I give you points on how well you use the terminology. That's important, whether you use the terminology, used it correctly, and you used it appropriately. And then kind of a last category where, as an instructor, have you demonstrated to me that you understand the concept? All right. So, for example, with this exam, it was evolution, right? Big thing we talked about in class. Well, all the chapters basically were about evolution, and it was important for me that you understand the mechanism of it. So that's why that question focused so much on what's adaptation, what's drift, what are the differences that occur, because that's very important in thinking about how evolutionary changes occur in organisms as they move along. So take a look at all that, read those comments. I do have some general comments, which is basically kind of the, I don't know, what do you want to call it? I don't want to say the optimum answer, but one good answer. Your answer might be a little different. You might notice, for example, that one thing that's in my answer is not in yours, and you're like, wow, he didn't ding me for that. Well, I might not, especially if you were really good at explaining something else. I'm looking holistically, all right? I'm looking at the overall idea, and that's how I had worded that question, right? Do you understand Darwin's theory? Do you understand it's about differential reproductivity? And do you understand that things can change, but they don't have to do with evolution, all right? So, and then, of course, look through all the multiple choice and all that. Make sure there's nothing wonky in there. I don't think there is. I think I've cleaned all that up. Anybody got any other questions for me? Our next exam is a little shorter, right? Chapters 27 and 28. So this is a good chance to, although you all as a class did pretty well on the exam, which I love. Nothing better for teachers than a great bunch of good exams, let me tell you. That's good stuff. So any other questions? Field notebooks. Hey, is anybody going on the field trip? I can't remember when the announcement. I have an announcement deadline for you guys if you want to go on the field trip. That's just so I can give the person who's doing it kind of a head count. Let me see what's the date for our first one. Is to Pear Park. It's a half a day, 9 to noon, and I know some of you work, so I understand that it's a matter of not being able to get there. But if you do have the opportunity, please, come on out to the field trip. It's a good time, good weather. Where did I say for? Let me know by the 13th. Okay, so we still got time. That's good. And then we're going to, the other one is at Lake Louisa, right? That's the one? Right. That one you need to let me know by the 27th. Okay, so we still got time. I guess everything's compressing. Announcements. Look in the announcements. There's two of them. There's one that says Pear Park, and the other one says Lake Louisa Hallowweed field trip. That's clever, isn't it? If you want to do the Hallowweed field trip, please read through the instructions. There's an app you have to download on your smartphone if you want to do that one with all of us. Someone else came in. Philip, how are you today? Good, good, good. All righty, so if there's no further questions or anything like that, I'm going to go ahead and get started on Chapter 27. Does anybody need to talk about anything with me before we go? Of course, if you need to after class as well, I can stick around and talk. I will be on campus tomorrow. If you do want to stop by for some in-person office hours, that's fine as well. Chapter 27, all right. One of the things that one of my little fascinations is electron microscope pictures, especially when they do things like they take an electron microscope or even just a microscope picture through a microscope of some ordinary object. I don't know if you've ever seen it, but sometimes it's like the wing of a butterfly. If you ever seen that under a microscope, you're like, whoa, that's really cool, you know. And then you'll understand why butterflies belong to a group of insects, an order of insects that we call the Lepidoptera, which basically means scaly wings. So there's a little hint, right? Anyway. So with that in mind, what I got here is a picture that I really like. It's false color, so these aren't the color of the actual items in the photograph, but it's an electron microscope picture. So you got to think about something being magnified a bunch. You might immediately, considering the chapter title, you might immediately figure out what the yellow things are. But what's the purple thing that they're mounted on? They're stuck on. It looks like it's a mountain. Or what is that? Does anybody know what everyday ordinary object is that? You do this online sometimes. I go, what is this? And you're like, no kidding, that's what that looks like. Especially when you get at that level, right? Nobody's got a guess from here. Now think something that's been magnified a whole lot. And you'll notice, and I'll give you kind of a little bit of a hint, notice that the mountain, well, it kind of comes to a peak, or let's say it comes to a point. Now, you might be able to think about something that, you know, if you magnify a lot, well, pretty close, Philip, yeah. And yeah, pretty close there too. It's actually the head, or not the head, excuse me, the pointy end of a straight pin. You know, kind of like you use in sewing, right? So this is your common everyday straight pin, and of course I already gave it away, didn't I? What's all the yellow things that are stuck all over it? Yeah, lots of them. Bacteria are everywhere, right? We did our plates last week. I looked at them just the other day, so they've only been incubating a couple days, and we got some good growth going on there. So that's the purpose of why I did that part in lab, is to demonstrate to you that these things are what we say are ubiquitous, all right? Ubiquitous is kind of a fancy way, if I can spell it correctly, ubiquitous. I don't know if that's it. That looks like Quados. Looks like a food. How do you spell it? Anyway, somewhere around there. I just spelled it the same way. Means it's everywhere. Things that are ubiquitous are everywhere. Bacteria, prokaryotes. Don't forget the prokaryotes include two domains. That's the bacteria and the archaeans. They split that up years ago from the kingdom Monera. But those things are found everywhere. So, you know, that's why we get this basis of germophobia, because they are everywhere. I've told you guys in class, or maybe I have, that the human body is a trillion cells, and they estimate that inside and on our body there's about 10 times as many prokaryotes. So your body is carrying 10 times as many prokaryotic cells. So you're mostly prokaryote. You're mostly this thing that prokaryote grows in. That's kind of interesting to look at that way, right? Now, so like I said, the basis for germophobia. But before we get too freaked out about these things being everywhere, remember it's kind of like silent mutations that you learned about in Bio 1. You learned about the fact that genetic mutations, and you even learned about it earlier on the semester with me. We did some number calculations. What was it? About 65,000 mutations a day occur among humans. But remember at the time I said that that's not as significant as it sounds, even though it's really not all that significant considering, 7 billion people on the planet. But remember, a lot of those mutations are silent mutations. And eukaryotes, that's going to be something, that's a mutation that's not going to show itself. So those kinds of things are, you might say mutations are bad, but in that case, this is a mutation that's neutral. And that's what a lot of bacteria are like as well, folks. There are the bad ones. They get all the press. They get all the glory. They get all the, oh, my goodness, it's going to kill everybody on the planet. But most bacteria, come on now, just kind of trying to live their bacterial lives and raise their bacterial kids and just do bacterial things. They ain't messing with you. They might even be beneficial. Helpful, right? Give me some examples of helpful prokaryotes. Are they all bad? They're not all bad. Come on. There's got to be good prokaryotes out there. Anybody like yogurt? How about Swiss cheese? My wife loves Swiss cheese. What makes the holes in Swiss cheese? Of course, it's folks in Switzerland with little cheese drills that drill those holes in there, right? Does anybody know what makes the holes in Swiss cheese? Come on, that's a question that I see all the time. What makes the holes? And maybe I don't see it all the time. Maybe I'm the only one. That's right. Yep. Oh yeah, that same thing. Absolutely, all of that. Oh, what you said, that's right, Philip. Those are actually gas bubbles produced by a bacterium that's been introduced to the cheese. See, bacteria, in that case, my microbiology teacher in college came from industry. What he did is he was from Wisconsin. He worked in the dairy industry up there as a microbiologist when he got out of college, right? So that would make sense because, of course, the dairy, cheese, all that sort of stuff, that's all using bacteria and stuff to cultivate these things, right? So there are beneficial ones. It's not just the bad ones. We have them in our gut too, Aaliyah, not just the cow, right? We got tons of what we call our, gut fauna, gut flora, which is a collection of bacteria in our gut, and we're learning a lot more about, having the right ones and the right mixture of ones. That's kind of the basis of what probiotics, isn't it? Having the right bacteria in your gut to do the right thing. See, those bacteria, those 10 trillion bacteria, they're not a bother, right? For the most part, they're leaving you alone. And there's another good thing about being covered in that many bacteria. Bacteria compete for space, right? Just like in evolution, just like when we're talking about Darwinian, there's more bacteria born that can possibly hope to survive, so they compete for space. So if your cover of your skin is covered with those 10, well, it wouldn't be the entire 10 trillion, but a lot of those bacteria, then and they're not bothering you. They're doing their thing, and you doing your thing. In other words, they're not really interacting with you in any sort of way you gain a benefit. You do get a benefit because they're taking up space of where some nasty bacteria could take a hold, right? Things we call pathogenic bacteria. Those are the ones that get all the press, disease causing. Now, we, of course, will talk about those, but I want to talk about the incredible uniqueness of these organisms. Things that you may not have ever thought about, and it's probably because they're small, right? Things that are small don't. If you looked on the paper yesterday and they said, "Oh my goodness, they discovered a new bacterium," you might go, "Yeah, big deal." But if they said they discovered a new species of whale, big things like that get all the press because we see them everywhere. We didn't even really know about bacteria until microscopes got good enough. But look at some of the things I've collected about how they can live. In other words, the conditions they can live in. And if you remember, our chapter 25 was the one, the history of evolution of life on Earth, right? Remember what the Earth was like really early on, right? Wasn't a very nice place. It was a very radical place. It was a very extreme type of place with a lot of hydrogen sulfide, noxious gases, hot thermal vents, you name it, it wasn't a great neighborhood at the time, at least as far as we're concerned. But we know life began back then. We talked about that. We see it in the fossil record, okay? But we got to remember that it's probably not living forms that are representative of most of the living forms today, though. We don't live in that kind of condition. That's something that you should automatically be knee-jerk by now, right? It's all about evolution. But look at this stuff. For example, there are prokaryotes who can live under an environment in which the amount of radiation, when measured in rads, is 3,000 times the fatal human dose. Fatal. We're talking about you get zapped with whatever 1/3000th of that is. I'm not even going to pretend to be able to do that in my head. You get zapped with that much, you're dead. But they can live in those kinds of conditions. They can live in pHs to the point. I'm thinking about Alien, one of my favorite movies of all times, by the way, where the blood of the creature dissolves metal because they have pHs just above zero. That's a nasty. The stuff in your stomach is 2, pH 2, you know? That's pretty strong stuff. Boiling is 100 degrees in Celsius, right? And they can live in temperatures practically right near boiling. And they can live in salinities, which is really something interesting because it's the drying out. And you remember drying out and losing water is an osmosis thing. There's nothing you can do about it. Water's going to leave your body in an environment. Oops. Sorry about that, folks. Water's going to leave your body in an environment that has more solute than you because it does. Just like a rock rolls downhill. I was explaining osmosis to my Bio 1 students today. So how do these guys deal with that? I mean, the ocean, the salt water in the ocean is about 3.5% solute. And your body cells, if you remember from Bio 1, is a little less than 1. Human cells have a solute concentration of just about 0.9. So that's why you dehydrate in the ocean, because it's got a higher solute. But these guys can live in environments that have, was that about 7-8 times saltier than that? How do they prevent drying out? These are extreme living organisms. Now, what I want you to think about is a couple of things I talked about in chapter 25 about where the origins of life came from. And one of the things I mentioned, or more than one, I mentioned the geothermal vents. Where do we find those geothermal vents? Does anybody remember? Where do we find those? Are they found in, your backyard? It'd be cool if it was, right? Oh, my God. Yeah, way, way, way underwater, right? Way deep. Remember, we didn't discover them, I told you, until, I think it was the early 70s, 72, 73, I want to say, right about that time. They're rare. As a matter of fact, they're so cool because they're communities of organisms that only, I know I've said this before, I'm sorry, but if I have, bear with me, that live around these geothermal vents, those species that only exist right there. They don't exist 10 feet away. They don't exist. So I am the only member of my species on the entire planet, and it's just me and half dozen of us other guys. That's incredible to me, okay? But one of the things I mentioned are one of those theories that used to be way out there, but remember, way out there theories, sometimes, people say maybe that ain't so way out there. Remember Wegener's continental drift? They called it rubbish. They called it junk. They yelled at him like the British do. Rubbish. The poor guy, can you imagine your life's work? Now come to find out, Wegener, he's right about the way those continents did that. Okay? So one of the things I mentioned was panspermia. And I said, there is a possibility, some people suggest, and not people from, the fringes, but some people in the mainstream suggest that relatively simple living things, like maybe these guys here, might be able to survive, or relative to these guys here, might be able to survive, or similar to them, planetary body travel, right? Like out on asteroids and things like that, and comets. Pretty nasty environments. Don't get me wrong. I'm not going to be one zipping around of the space on an asteroid. It's cold, and you're getting radiated and all that sort of stuff. But if you were a critter, that wasn't a big deal to you, you might be able to survive that. Now, if that's intriguing to you, let me show you a piece of little evidence here that occurred. Oh gosh, this is relatively new. I think I was teaching in Valencia, so I think in the 90s. They discovered a meteorite in Antarctica. Okay? No big deal. Well, it is a big deal. Meteorites are cool. But for reasons that have to do with understanding geology, in the same geology class, but for reasons of geology, the folks who looked at this meteorite have hypothesized or have data to suggest that it is of Martian origin. The meteorite is. In other words, the rock came from Mars. Now you're thinking, how the hell did the rock get from Mars to Earth? Well, I don't know. Mars gets hit by something, right? An asteroid or a planet or meteorite, or some little bigger than that, throws up a bunch of stuff up into the air, like you can imagine would happen. Most of that stuff falls back down to Mars, but some of it might get far enough out there that it could actually leave the orbit of Mars and become interplanetary. So it's out there between Mars and Earth for God knows how long, right? Millions of years, and then it finally gets close enough to Earth, and Earth's gravitational attraction kind of sucks it in, and eventually it spirals down and it comes to Earth as a meteorite, right? That, in a nutshell, is the science of all that, right? rocks flying around down there in space. Now, what's significant about this is not so much that it came from Mars. I mean, that is kind of significant, but we do have other meteorites that came from other places, we believe. I believe some other Martian ones. But if you look in the picture, what we're looking at looks like, I don't know, okay, whatever it looks like something that was formed in a regular fashion, doesn't it? You know, if you look at rock forms, I get a crystalline form and all that sort of stuff. But if you look at that thing in the middle, doesn't that almost look like it's something that really you would almost expect to see among living things, right? It almost looks worm-like, doesn't it? Agreed? Right? Thumbs up, thumbs down? What do you think? I mean, we're all scientists here. We can do that. Yeah, I would. That certainly doesn't, if you're looking through rock, and this is with an electron microscope, if you're looking through rock and you saw most of that other stuff that you see around those lumps and bumps, and you came across something like that, and you go, that's weird. Now, what is that? Well, okay, we're not sure what it is. Could it be fossilized bacterium? It could be. It could be. We have fossils on Earth. Remember when the first earliest fossils were 3.5 or 3.6 billion years ago, that have fossilized prokaryotes. They don't fossilize very often, soft-bodied critters, right? And they don't fossilize very well, but every once in a while you get lucky. So, and this looks similar to the fossilized bacteria that we found on Earth. Now, in fairness, there are other folks, geologists, for example, that suggest processes geological that could form this kind of structure. So it could be just a trick of the eye. But it may not be, right? So the whole idea is, if this rock came from Mars, is this a fossilized evidence of Martian bacterial life? And if we go to Mars and we explore it for God knows how long, and we never find life on it, which I don't think that's the case. I think we will. I think we will find prokaryotic life. We may have the only record right here that life ever occurred on Mars. I think that's kind of neat. Would you go to Mars if they gave you the chance? Go live on Mars? I think it's like, 6 months to get there. Heck yeah, I would too. I'd love that. All right. If we're all sufficiently impressed by these critters, and hopefully you are, then let's go ahead and start digging into the structure of them. Oh, no, that was the one with the potatoes. That was the one with, come on, who's his name? The actor. He grew potatoes in The Martian. Isn't that the movie where he grew all those potatoes? He was the last one left. Yeah, right, remember? What's his name? Why is it eluding me? All right. What do we know about little prokaryotic cells? Well, you guys know some already. You've learned some in Bio 1. You learned that for the most part they're unicellular. But I want to explain to you that as we go through different groups of organisms, I'm going to talk to you about the general characteristics of these critters. But I'm also going to throw in the fact that there are those types of critters within these groups that push the envelope, that sort of go between the rungs of Aristotle's ladder, that sort of do a little of this and a little of that. So if you say, are they one of these or are they one of these, the answer is yes. It's kind of like the definition of a species, right? Organisms that are hybrids. Where is this in a species and where is it not a different species? So among this whole idea of unicellular prokaryotes, even though most of the times they're unicellular, there are some species that get together. Temporarily sometimes, and play cards, and bet, and get drunk, and then the cops come. See if any of you are awake. Now, why would they form colonies? Because for one thing, life in a colony, there's an advantage. There's an advantage in numbers. Like, for example, a colony of you, like you see in that Petri dish down below you, that's kind of what we're going to be seeing next week. A colony of you might do a better job of digesting a certain nutrient. Maybe if you divided up the work, you could get it done faster. This is really nothing too spectacular, right? If you think about it, humans, we do the same thing. evolutionary biologists tell us that humans as a species would have never survived had we not learned to be social. That as a group, we're very powerful, but by ourselves, we're relatively weak. It just reminds me of the Planet of the Apes movie. One ape weak, many apes strong. I love that because that's exactly what we're talking about here, right? So sometimes these organisms can form colonies, sometimes, like I said, temporarily, moving in and out, depending. If the environment is rich in nutrients and it's a bonanza, everybody's living on their own. They don't need each other. But man, I tell you, when stuff gets scarce, it might be beneficial to start living together. And even living together, you can start dividing up the jobs. So what am I getting at here? These are one of those in between the rung ladders. And you're thinking, well, what does he mean? Because when we get into the fungus, the plants, especially the animals, this whole idea of multicellularity becomes very interesting. See, multicellularity is a little more than just cell. There's no multicellularity in prokaryotic cells. They're all individual organisms, not part of a greater organism like your cells. And that's a big difference. Animals took that whole idea of multicellularity, man, to the next level, like on steroids. That's why we've got animals have such complex motion and complexity to them in general, right? But I just want to show you that this whole idea of kind of like what would be the evolutionary advantage of cells sticking together. We can see advantages of that, or we can see evidence of that way back in the prokaryotes. Forming these colonies might have been better. And of course, we talked about this the other day, so I don't need to spend a lot of time, but they differ on shape. We looked under the microscope. One of the, what the one I forgot to tell you about was when they're in pairs, two round ones in a pair is a diplococci. That's important later on. We'll look at some of the pathogenic ones, and a very, what's the word? No, infamous, not famous, but very infamous diplococcic bacterium is the one that causes gonorrhea. So that is a 2 circle. So make sure you know different shapes, right? Size. They're small. Now we know why they're small. This is Bio 1 again. Why do cells stay small? Why don't they get big? I mean, some prokaryotes can get, well, what's this biggest one? Thiomargarita nambiensis. Thio means sulfur, so it has something to do with sulfur, 750 microns. But for the most part, 1 to 5 microns in prokaryotes, 10 to 100 in eukaryotes. Why do cells stay small? Remember why? Oh my goodness. Back to your Bio 1 teachers. Did I have any of you for Bio 1? I don't remember. Way back in the early part of Bio 1, when you talked about cells in Chapter 4, the first thing you probably talked about, or maybe it was Chapter 4, the first thing you probably talked about is why they're so doggone small. Nobody remembers why cells stay small. There must be an evolutionary reason. That's exactly right. When they're small, their surface area to volume ratio is higher, so they're much more efficient at exchanging nutrients, exchanging materials. All right, now prokaryotic cells take that to the extreme. They're really small. See, 10 to 100 microns in a typical eukaryotic, your cheek cell that you measured under the lab, under the microscope. We just did it the other day with my other Bio 1, 40 to 50 microns, so it runs right about in the middle. Even some of your smaller ones are bigger than some of the bigger prokaryotes. Blood cells are about 5 microns, so they're kind of at the upper end, but nonetheless, they stay small, other than that big one you see there. The little b, if you wonder why it's in the pictures, to give you some sense of scale. And it's not that Thiomargarita is that size compared to a bee, but you see the dot on the bee. That's the Thiomargarita bacteria. So for the most part, they stay small, but there are the extremes of that, right? Cell walls, cell surfaces, man, these dudes are all about cell walls and cell surfaces. Not the same type of cell wall in a plant cell, first of all. Let me make that clear. It's not made of cellulose, okay? Nearly all prokaryotes have these, but even though it's not made of cellulose, it does a lot of the same stuff that cellulose did for plant cells. So this is like really major convergent evolution, isn't it, right? We've got prokaryotes coming up with a solution that plants did later on by building a wall. Maybe it's not made of the same material, but it maintains the shape of the cell, right? Keeps it kind of square. It's physical protection, and there's some physical protection by a cell wall of a plant cell too. And then the last one, right, number 3, cell walls are really important in plant cells because remember, plants can't go get water, can they? They have to store it. They store in that big central vacuole, which swells. Now, if you're an animal cell, water moving in to the cell is good until a point, until lysing, right? But plant cells, I always tell my Bio 1 students, plant cells are like trying to blow a balloon up in a box. You can only blow it up so far. So that wall helps those plant cells be able to take on lots of water because, again, they need to store that. Now, not such a storage thing in prokaryotes, but they live a lot of times in hypotonic environments. So this prevents them from bursting, the cell wall that they have. Again, different structure, but same function. See what I mean by convergent evolution at a really wide level, right? More than one way to skin a cat, Mother Nature says, which is horrible, especially if you like cats. Let's talk about that cell wall. Let's see what's so different about that, okay? Among prokaryotes, among bacteria prokaryotes, that domain, there are 2 very basic types. And if you go on and you take micro, these words, these terms will become very familiar to you, and they become the basis of identifying. See, it's a lot easier with big critters to identify them. But when they're little critters, they're like these prokaryotes are a lot more difficult. So sometimes you use a lot of stains and tests to do that. If you look down below in the picture, I've indicated, or they've indicated, with gram-positive and gram-negative bacteria. Now, what the gram is a stain. It's called gram staining that you do to these organisms. I'll show you how that works in just a second. But it's very easy to tell gram-positive bacteria from gram-negative bacteria because of their color, right? Gram-positive bacteria under the microscope are purple. When they're stained gram staining, you can make them purple with other things, but when you gram stain them, and the ones that aren't gram positive are reddish pink in color. We call them gram negatives. All right, now I'll talk in just a minute about how we do a gram stain, and then I'll relate that to the structure of the cell membranes of gram positive and gram negative. But I want to go to that first. I don't want to show you the staining process, excuse me, quite yet. All right, start with what we know. Look at the gram positive. First, we got a plasma membrane. All right, nothing unusual there. Plasma membrane is made up of phospholipids and a bilayer, and it's got embedded proteins. And I'm hoping this is all very familiar because it's all Bio 1 stuff, right? Where it isn't Bio 1 is the cell wall. And it's not even Bio 1 in terms of the plant cell, because notice the cell wall is not cellulose. It's made of this really, in science in college, one of the things you do is you learn big words. Peptidoglycan. Who was the other day we did it in class? Abu Defduff. Remember I said if you don't know how to pronounce a word, or was I doing a Bio 1, a biology, and many times they're very phonetic. So that's peptidoglycan. So peptidoglycan is a mixture of peptides, proteins, of sugars. Well, the point is it makes a thickening, a cell wall. If you're a gram-positive bacterium, you basically got your plasma membrane and your cell wall of peptidoglycan, all right? If you're gram-negative, you got a sandwich arrangement. Gram-negatives have 2 phospholipid bilayers. See that? In between is a much thinner layer of peptidoglycan. The outer membrane and the peptidoglycan, both of those make up the cell wall of gram-negatives. As a matter of fact, gram-negative bacteria many times tend to be the ones that cause disease, and I don't know how well, I can't anyway with my glasses. If you look at the picture, those little green hairs coming out of the top of that outer membrane, those are lipopolysaccharides. So we got lipids, and then we got sugars stuck together, and many times in gram-negative bacteria, what makes you sick if you have a bacterial infection is the endotoxins that these lipopolysaccharides secrete. So you can see from a medical point of view, it would be interesting, right? You would test a patient with symptoms, a symptomatic patient, to see if they were infected with a gram-positive or gram-negative bacteria. Now, gram staining is very easy to do. If you take micro, you'll do one, and like I said, I'll show you in just a minute. So it's a real quick and easy test to do. Now, the reason why they're purple or red when they're gram stained has to do with the structure. So before I get to that, let's take a look at it. The gram-positive bacteria has a thick peptidoglycan layer, while the gram-negative has a thin. Okay? So think about the thickness and the thinness of those relative peptidoglycan layers. So here's how we do a gram stain. Throw a bunch of bacteria on there and we apply a dye called crystal violet. Crystal violet is a common dye. We have some in our lab. So we throw that on there and everybody gets stained purple, right? Gram-positive bacteria are purple. Gram-negative bacteria are purple. Now, the next thing we do is a little trick of chemistry, but they throw something called Gram's iodine. If you're wondering about the Gram, I think it's probably the dude who came up with this, right? So you throw a little bit of that Gram's iodine on there, and the Gram iodine forms a chemical matrix, a complex with the crystal violet. So what that basically does is it solidifies the stain, the purple stain inside of the cell. Now, the peptidoglycan layer, the thinness and the thickness, comes into play in the next step. Because the next step, you use alcohol and you apply alcohol. Now, what alcohol does is it causes that peptidoglycan layer to shrink. Okay? And here's where the difference lies. When you shrink... Hold on a second, please. Stop. That's Cooper and the UPS guy, I think. When you shrink the thick peptidoglycan layer, right, what it does is there's enough of it there, so when it shrinks down, it's like shrink wrap. Hold on 1 second. Let me get him this other guy. That's enough. He's gone. Come on, chill out. Anyway, so it's like shrink wrap. So that thicker layer traps the dye inside, but the thinner layer in the gram-negative, it shrinks too, but it's much thinner. So it's like a thin shrink wrap. So that peptidoglycan layer in the gram-negative one is not thick enough to trap the dye. So when you come back with the alcohol, what the alcohol does is it shrinks that peptidoglycan, that's the PG, right? Shrinks that peptidoglycan layer, traps that dye and that iodine inside. In the gram-negatives, the peptidoglycan layer shrinks, but it's so thin, it doesn't prevent the alcohol from then washing the stain out, right? So the alcohol has a little tiny effect on that peptidoglycan layer as far as the size of it. So when it shrinks, there's not enough, and the alcohol washes the dye out. So right away, you got your difference between the two. Now, the reason why they come behind at the last step and dye, we used a saffron red dye, is just so you can tell gram-positive from gram-negative. Saffron red actually dyes both of them. But the purple ones, the gram-positive ones, are already so dark purple that you can't see the red dye. And then down below, once again, you can see an example of how beautiful this is, right? You'll do this in micro, trust me. I know you will. Very simple staining technique, bam, bam, bam. We know gram-positive. And we know gram-negative. And like I said, from a medical point of view, that might be important because a lot of the nasties out there are gram-negatives. So this is a whole colony of them. It's like one of those things that I used to look at the mall years ago. Do you remember those? When you look at the mall and you would let your eyes kind of go out of focus and then you would see a bridge or a lion or something like that? I never could see those things. But you can easily pick out the gram-positives and the gram-negatives in here, can't you? How many of you all have ever had strep throat? I had it when I was a kid. It's nasty. Remember we talked the other day in class when we were looking at the shape of bacteria, their names. Now you kind of know what it means when we talk about strep throat. We're talking about streptococcus. So streptococcus is going to be the organism. So think about that. If you were in a medical field, you would be, if you want to identify it, I'm sure they don't do it through a microscope, but you would expect to see these organisms, these streptococcus, these things that cause strep throat, to be circular and be in a straight line, to be a streptococcus, right? This is actually a picture of a strep throat cell, a streptococci. Actually, coccus. I'm sorry, cocci is plural. So this is a streptococcus organism, and you can see that inside is the nucleoid region, the chromosomes, and all that sort of stuff. But what's sinister about this picture? There's a cell wall, right? But there's also an additional layer, right? This whole section I'm talking about is cell walls and surface structures. So in Streptococcus, they have the bacterial cell wall. Then on the outside is a slime layer. a bacterial layer, or just this is layer of material that does a couple of things. What it does is it allows that cell to stick to, right there's some picture, to a tonsil cell. It provides protection against this cell drying out. For some reason, I'm having a hard time with my throat today. Hope I'm not getting strep. Great, now that I talked about it. I probably will. Hi, Patrick. How are you today? We were just talking about strep throat. And then another thing that this capsule or slime layer does is it hides that cell. You see, your immune system is a series of cells that go around. I'm doing good, thank you. That go around and basically check for cells, whether they're supposed to be there or not, whether they're foreign or not. And I'm sure you remember from Bio 1 that has to do with immune system cells recognize the outer glycolipids and glycoprotein structure of the cells in your body. That's how they recognize by shape. Well, if you can hide that with that slime layer, like the Streptococcus is doing it, then you're basically hidden from the immune system. Immune system is not gonna know that you're hiding. You're gonna be, woof, I'm invisible, even though you're really not invisible, but you are to the immune system. This is Bacillus anthracis. Anybody know what Bacillus anthracis causes? Bacillus anthracis causes? Yeah, 3 times real fast. There's a disease out there that oftentimes, especially with those that get all the bad PR, we have a disease we name after the critter that caused it. You've heard of anthrax, right? This is the critter that causes anthrax. Right after 9/11, which I think probably a lot of you were rather small, but there were these scares. People were somebody was mailing anthrax spores to people. Now, that's my point here. Another one of these surface structure modifications is some of these critters, and this goes right back into our earlier chapters, can produce this very resistant endospore. That's what they're pointing at. The whole outside is the outside of the critter, but they're producing this endospore, and they produce that during times when the conditions or the environment is not conducive to their survival. Now, what these people were doing after 9/11 is the military has biological weapons, and one of the things they have is spores of anthrax, this critter. Drop them on the enemy. What causes them to germinate is moisture. So if you inhale them into your mucous membranes, into your lungs, they would then germinate. They produce a toxin, and basically it can kill you, or it does kill you, or it's very, very often fatal. The organism is producing these endospores because the environment is not conducive. So this is this is exactly what we talked about in evolution. Remember, if organisms don't adapt to their environment, then they go extinct. But there is a workaround. There is a loophole. If you can produce something where you can protect yourself from the environment, like this resistant endospore, you can wait it out. You can wait it out. You can wait until the environment changes. Oh, gosh, it's getting dry. It's getting dry. We need to move, George. We need to move, George. No, George says, I'm just going to make this endospore. I'm going to go into this, and it's a lot deeper than hibernation. Are endospore things dead? No. If they were dead, they wouldn't be able to come back alive. Are they alive? Barely, yeah, right? Metabolically inactive, we say. So they're capable of turning it all back on again, okay? But isn't that a great technique? Isn't that a great loophole? If I don't like the environment, now keep that in mind, because other critters, when we're going to talk about, do the same thing. Like fungus do the same thing, plants do the same thing, even animals. They all have these certain species that can form this weighted out structure. The heck with the environment or me adapting to it. I'm going to wait around until the environment adapts to me. It's kind of stubborn, but if you can deal with it, then why not? Yeah. Imagine if we could, right? Absolutely. Isn't that the whole idea behind freezing? Like cryogenics, freezing folks if you suffer from something so that years later I don't know if I want to come back years later, though. You know what I mean? Maybe live 100, 200 years, but I don't think I want to die or be frozen and come back in 100 years. I don't know about that. That'd be a little weird. Might be too much shock. What else can they do with these things? Well, just like I showed you with the Streptococcus, what they do is they can make these fimbriae. And fimbriae are basically extensions out of the cell surface again, which they use, and many times they can secrete a little bit of adhesive, but just the overall nature, they can kind of grab onto stuff. Kind of creepy to think about, right? Fimbriae also have another purpose, like you see in the upper right. The organism in the lower left has extended my fimbriae out to you. It's extended its fimbriae up into the organism in the upper right long enough so now the fimbriae has become something we call a pili, and a pili is a tube by which these organisms can actually exchange things. And these guys, what they're specifically doing are exchanging DNA. So it's a sex pili. So there they are. That's the naughty bits of prokaryotes. That's them in the middle of it. So and they can exchange DNA. DNA in a variety of ways. Obviously, I get excited now. I stretch my throat. But this is one way they can do it. By forming that. Sorry, folks. My goodness. All right. We'll talk more about this whole idea of exchanging DNA in these guys a little bit later on. Let's talk a little bit about how they move around, or what we call motility. About half of them can do that. Some can move pretty fast. Some of them have been clocked scale, right? Scale to their size, but is it similar to us running at 90 miles an hour? Like the $6 Million Man, right? But he ran at 60 miles an hour. So some of these guys have someplace to be pretty quick, obviously. Most common method to move around is a flagella. Now, you were introduced to a flagella in Bio 1, and there's a flagella in this organism as well, but that's unique. I want to wait just a second. I don't want to give this away. I want to see if I can lead you into this. All right, so they can use a flagella, and it works just like the flagella that you saw in Bio 1 with the eukaryotic cells, but this is a prokaryotic flagella. Spirochetes, the spiral ones, spirochetes another name for them, they move by basically just kind of twitching back and forth with these helical filaments that are right underneath the cell wall. Some of them have these slimy threads they can secrete, they can kind of pull themselves along, and they can also use that to stick to things. But I want to go back to the flagella for a minute. Cool little story. In the upper left is the prokaryotic flagella, and in the lower right is the eukaryotic and the prokaryotic. It's a different picture, but look at the eukaryotic for just a second. Let me refresh your memory. In Bio 1, you learned that eukaryotic flagella are made up of extensions of microtubules. You remember there were triplets of microtubules with motor proteins, and the thing wiggled back and forth, and it caused this thing to move around. And you might have remembered that flagella and cilia are identical in structure. They're both made up of microtubules, but the big difference is one's bigger and longer, the flagella are, and cilia generally they're shorter, smaller, and critters generally have lots of cilia, where they might only have a few flagella. All right? So that's cool. Now we know what the flagella do. They move the organism around. So now take your attention to the upper left to look at that. This is a prokaryotic flagella. Notice there's no microtubules, there's no motor proteins, there's no tubules sliding back and forth. That's what makes the eukaryotic flagella flex. The microtubules slide back and forth. But even though that is doing the same structure, but there is this really interesting rotary structure down below, and that's a really cool picture through the electron microscope. Isn't it neat? Nanotechnologists love this stuff because this is like little miniature machines. Now, let's stop right there for just a second. Notice that the structure of the flagella in the prokaryote is very different than in the eukaryote. But whether it's shaped or constructed the way the prokaryote is or the eukaryote is of one, they both do the same thing. What kind of structures are these? What's that name we use to refer to structures that have the same job but are very different in terms of how they're put together? get everything from earlier, did you? What's that called? There you go. And analogous structures, homologous structures would be same structure, different purpose. Homologous would be like a whale flipper, dog, paw, okay? Remember, homologous is going to refer to same origin. So the reason why the whale flipper and the dog paw are the same, we both had a common ancestor. All right, so Aaliyah says those are analogous structures. Now you tell me, through what type of evolution, what aspect of evolution did these two structures end up looking the same? What caused that to happen? Yeah, good. You're following me. Now, if you look down below, I've got a word that we introduced you back in evolution. The word is acceptation. Remember, feathers, legs, and eyes were acceptations. We asked the question, what good is 5% of an eye? What good is just part of an eye? Well, nobody says part of an eye is any good, but you could start off with pigment cells, and then you can make them into a cup, and then you can put them in a lens. Remember when I went through all that? We called those things acceptations. Who remembers why birds have feathers? It helps that they fly now, but that's not originally why the reptilian ancestor of birds. That's right, it's warmth. Feathers became fortuitous for another purpose later on, flying. The first feathers you remember in birds, this is great stuff, man. I love this evolution of birds, were not flyable feathers. They just kept you warm. Feathers have to have a certain shape to be able to fly. We call those acceptations because, like I said, they were produced for one very immediate purpose, warmth. Be able to tell light from dark, and through evolutionary changes, they became fortuitous, helping birds fly, eventually making a camera-type eye. Now, if you're wondering where I'm going with this, what I'm pointing to in the red, if you look at that, now look what the arrows that are indicating that protons are flowing through there. I don't want to give it away. This flagella spins. What causes the flagella to spin are protons moving through it. Protons are the force. As those protons diffuse through, as those protons move from this part right here to in here, following the arrows, they move through a mechanism that makes that rotor head spin, causes the flagella to spin. Have you seen this before? Not in this class. Have you seen this before, where protons moving through something made something spin? or turn. I don't. Maybe it wasn't spinning like a flagella spins. Maybe it is. I don't know. Have you seen this before? And where? Go back in your brain. Bio 1. Wasn't there something in Bio 1 that did this? And specifically, what part of the electron transport chain, Aaliyah? What part of the electron transport chain was set into motion, turning when protons flowed through it? It was one of those proteins that are found in the membrane, and specifically, it had a very similar structure that you see here. There you go, ATP synthase. Remember how ATP synthase worked? It worked just like she says. ATP synthase worked just like you see here. Protons are moved across the membrane in the mitochondria. They flow back, and they also in photosynthesis too. They flow back through the ATP synthase, turning the rotor and the catalytic knob of that ATP synthase to make ATP. Therefore, this spinning rotor thing is an acceptation. The spinning rotor thing evolved, all right, and then later on became fortuitous for things like spinning flagellas, probably initially, and creating boatloads of ATP using a spinning way of doing it, like they do it in the electron transport chain. right? Look, even down there at the cellular, molecular level, we see evidence of organisms who have come up with similar ideas through different steps, as well as set certain baselines of how to make things happen. Like when we invented the wheel. Oh, if we want to make things go, we put wheels on them. Put round things on things, and it's easier to push them. Mother Nature said, wow, I can make protons, because they want to diffuse by themselves. They want to go from that greenish membrane back to the other side, because you got more on one side than the other. That's just natural. Water rolls downhill, bricks fall off of a cliff. So if I can take advantage of that, a living thing, can pump those protons and make them flow back to that rotor. I can design that rotor so it'll spin. And I can spin a flagella, or I can make ATP. Cool story. Excitation down there at that level, right? So they can move away from things. They can move them toward them. We call that taxis, like a taxi, right? And if you're moving away or toward chemicals, it's called chemotaxis. If it's light, like there are many prokaryotes that are photosynthetic, that would be important to them, like they'd want to swim up in the lake. And some of them really cool can actually move in relation to the magnetic fields on the Earth, geotaxis. That's kind of neat, because a lot of these critters actually have little particles of iron in them that line up with the magnetic fields of the Earth and let them know, where that is, and that would be important, especially in a lake environment or ecosystem where these prokaryotes were actually wanted to go towards the bottom of the lake. How do they know top from bottom? Well, if they're phototaxis, that would help. But if they're geotaxis, the magnetic field changes as they were going down into the depth of the lake, and they would want to go down because that's where the nutrients are. So these things are helping them find ways to go. It's just kind of unique. There's even some thoughts that, well, I think there's some research to suggest that birds and fish use magnetic or iron particles in their body to actually line themselves up with the magnetic fields. So that's really kind of cool. Okay. As you remember from Bio 1, there really is not a lot to these guys in terms of cell organization. That doesn't mean there's not a lot to them in terms of complexity, but they don't got a lot of parts. They don't have a nucleus. There's no infolding. There's no internal compartmentalization. That was one of the things that eukaryotic cells were really well known for, folding in on themselves, engulfing things in membranes, that sort of stuff. There is in many of them folded membranes, like you see in those two pictures there. The aerobic prokaryote and the photosynthetic both have membranes, a respiratory or a thylakoid membrane. Back to the ATP synthase for just a second, an electron transport chain. The key to what makes that work is the membrane, is it not? You're not going to get protons to flow back through the ATP synthase or the little prokaryotic flagella unless you can create a situation where there's more on one side than the other. Now, by definition, and I know this seems obvious for me to say, but think about what I'm saying, you got to have something, some physical thing that allows you to create more on one side of it than another. So the basis of the electron transport chain is moving protons across the membrane and then allowing them to go back through ATP synthase to make ATP. You got to have the membrane there to move them across. So in the respiratory membrane of that aerobic prokaryote is what we find in that membrane of the mitochondria, and in the thylakoid membrane, the photosynthetic prokaryote, we find the same membrane we find in the chloroplast. So regardless of whether we're looking at it in the prokaryote or in the chloroplast and the mitochondria of eukaryote, it's all about the membranes, folks. It's all about moving protons over a membrane. Yeah, the reason how your cells conduct electrical impulses, the nervous system, is about things being moved across the membrane. Membrane. It's all about that, all right? Pretty simple genome, not nearly as big as ours, about 1/1000th as much DNA as we do, but now to qualify that, all of their DNA is exons. There's no introns. There's no intervals, right? It's all readable DNA. Remember, we got most of us eukaryotes, or a lot of it is introns. They're all exons. It's all one big loop. You could pull it out like you see in the picture there on the lower right, the thing that's labeled chromosome. That's actually, if you could follow it, one loop of DNA all the way, all connected into a big circular loop. A lot of them, oh gosh, this is cool. A lot of them have these accessory loops. It's labeled there, like little chunks, looks like they came off. They didn't. They're little pieces of DNA called plasmids. And the plasmids are like accessory pieces of DNA with usually just very few genes. They can provide for things like antibiotic resistance, special contingencies. Oh, a new type of food in the environment, a new type of environmental pressure in the environment, something that maybe comes and goes every once in a while. Those plasmids can not only make copies of themselves inside the cell, so they can replicate independent of that big thing, but here's something kind of cool, antibiotic resistance. If I'm a prokaryote and I have antibiotic resistance, and the reason why I have that is because there's some genetic information in my plasmid, and you're my buddy prokaryote, even if you're a different species, and you don't have this genetic information, I can give it to you. I can form a sex pili, if you're willing. I can form a sex pili between you and me, and I can give you that plasmid. So you don't have to have inherited, they don't really inherit it that way, that genetic information from your parents. They don't really have parents. You don't have to make a mutation to get that to happen. I can just give it to you. So from a medical point of view, that's kind of scary. Well, not scary, but alarming. That means that antibiotic resistance doesn't have to do with just creatures or organisms replicating and reproducing, and most of them die and some of them live. That is obviously what does happen as well. But once there is that antibiotic resistance genetically, especially in a plasmid, I can just give that plasmid to somebody else. I don't give it, I make a copy and give it to you. Isn't that neat? I'd like to have some bird plasmid so I can fly. All right, so these guys have really interesting ways of living, but one of the things that they can't get away from is the fact they have to be different. They have to be variable, and we talked about that. And these organisms, there's some more of them down there in the naughty bits right there. There's a sex pili between those two. These organisms have to have variation. That's just a rule of natural selection. You can't do anything about that. You got to have variation because the environment changes, but they don't have sexual reproduction. And you remember when I talked about variation, I said one of the biggest ways to generate sexual or variation was chiasma, crossing over that occurs. And the whole mixing up thing that occurs during meiosis anyway helps create variation. Well, these guys don't have that. They don't have vertical gene transfer. Vertical gene transfer is you passing your genetic information off to your offspring and getting it from your parents. Horizontal gene transfer, that would be like plasmids being passed back and forth. So where does this variability come from if these organisms don't have that chiasma, that crossing over, that meiosis, which they don't do any of that? Well, number one, mutations can arise spontaneously. Radiation can bring that on. Chemicals can increase that. Just the whole doggone process of DNA replication can hit a bump every once in a while. I mean, you learn to get it again about it in Bio 1, it unzips and then A to T and G to C, but that might not be exactly every time. Plasmids, like I talked about, mutations can very easily spread through a population. For example, Staphylococcus aureus, which is a scourge in hospitals. Hospitals deals with staph infections all the time. You think staph infection, you think, oh, infection among the staff. No, it's STAPH infection. And Staphylococcus aureus, specifically a strain of it, became resistant to something called methicillin, which is, you think about it, looks like the word penicillin, because it was an antibiotic. It was the thing to kill Staph aureus, and hospitals were using it like crazy. And now you know what that does. That actually furthered this whole idea of evolution and resistance against it. So again, they don't have a vertical gene transfer, but there are ways they can transfer information back and forth. Like I said, with the plasmids, and three other ways they can do that is through something called transformation, which is they actually take up pieces of DNA from the environment. Even from other species, they can take up pieces of DNA. So they're moving along. When you die, doesn't mean your DNA is not there. So these organisms could come along and pick up DNA from organisms that have ruptured in the environment and pick up, oh, there's a good gene, I'll use that one, They can do conjugation, that's the sex pili thing, from the little tube between them, so they could be exchanging a plasmid, or they can be exchanging their main genome or parts of their main genome. right? That'll do it. So that's basically mixing up the genetic information between two critters there. And then viruses can act as a vector. They can actually move genetic information from one to the other. So these guys have plenty of ways to come up with variation. Like I said, one of the things they can do is transformation. So a cell bursts, chunks of DNA are out there in the environment. Those blue chunks, along comes another cell, picks up that piece of DNA, and as you see, incorporates it right in there. This occurs naturally. It's what you studied or what you learned about when Griffith was the fellow who was injecting the rats with the R strain and the S strain of the Streptococcus pneumonia. You remember where he injected the living R, and then the heat killed R or S, and then the dead. You remember all that in Bio 1? That's how the change was happening. That's why the mice, when they were injected with the virulent strain, but it was dead, when they died and he necropsied them, when he cut it open, he found living virulent strain. Now, he didn't put that in the mouse. Remember, he killed the virulent strain. He put the non-virulent strain. But the non-virulent strain, the one that doesn't kill the mouse, picked up the DNA from the dead virulent and it became virulent itself, and that's what killed the mouse. That's transformation. That was natural. DNA can be picked up even from a dead cell by another live cell. We do it in the lab. We put DNA into the environment of E. coli. They naturally, we do it artificially, they naturally would take up that piece of DNA, and then we can clone those E. coli, make millions of them, and we can use whatever gene we stuck in there to make something. Like, for example, insulin. The hormone necessary for regulating your sugar in your blood, diabetics, right? We actually can take E. coli cells, put in the insulin gene, imagine in this picture the blue, and then E. coli has no use for insulin. So every time it's reading its DNA, now remember, it doesn't have any introns, so it reads it all. So you stick in you stick in artificially that insulin gene, and it goes through and it reads all the genes it needs to make E. coli stuff, and blip, makes insulin. Now, since it doesn't need it, it exports that insulin out of the cell, and you grow up these E. coli in big vats, and you make boatloads of insulin. They got all kinds of molecular machinery now by which to actually, you can write your own little DNA fragment. So it's going to get to the point where we don't even need the cells to do this anymore. We can just sit down. What do you want? Oh, okay. We type out a genetic code. Boop, there it is. Maybe use a 3D printer and you print up something. Can you print me up a Tyrannosaurus? Why, sure. Hold on. Maybe not so good. Conjugation with the pili between them. Look, here's how they do that. So here's one passing a plasmid to the other. The plasmid's the part in the orange up above. So we're going to transfer, in this case, it's called an F plasmid. And the whole F plus and F minus is very simple. F plus, you see this used a lot. F pluses, they have it. F minuses, they don't. Okay? Now, the F plasmid, what does it contain? It has genes on it. I'm not really sure. But let's say this was the gene for being methicillin resistant. So a couple of these Streptococcus, now these aren't shaped like Streptococcus, but go with me on this. The one guy on the left says, hey, I got the genes for methicillin resistant. The F minus guy says, give that to me. I can't give it to you because then I would lose it, but I'll give you a copy of it. So it unrolls it. Oh, this is so clever. It unrolls it, and half of the DNA is passed to the other guy, to the recipient. Only half, one half of the latter. The guy who receives it, the guy, I keep saying guy, whatever, the organism that receives it makes the other half. That's so easy to do, isn't it? ATGC. And then the half that was left behind in the donor cell, he completes it by doing the ATGC and forming the other half. And in the end, they disconnect their 6 pili, boom, that plasmid's been passed. Happens in just a matter of short period of time. Devious little critters, aren't they? Mixing up their genetic information. Now, these aren't critters. Well, the green thing is a bacterial cell, and all of the little orange things stuck all over it are a picture of the thing you see in the drawing are viruses. Now, viruses are weird things. Are they alive? No. Not by the true definition of being alive. They can't reproduce on their own. They need a host cell to do that, and that's one of the characteristics of living things is reproduction. But what they use that host cell for, if you look in the picture in the upper right, is they carry along pieces of DNA, that viral DNA, and they inject that into living cells. They can inject it into your living cells, and in this case, this virus is actually injecting it into an E. coli. These now viruses have all kinds of shapes. These guys right here that look like these future spacecraft critters are actually called, I'm typing it, bacteriophages, to eat bacteria, to destroy bacteria. So they inject that viral DNA into the bacteria, and that E. coli does with that viral DNA the same thing it would do if we injected it with insulin genes. But instead of making insulin, it makes viral genes. It makes genes necessary to make more viruses. So the phage, the bacteriophage, attacks the cell at the top, right? It injects its DNA into it. That causes the DNA of the virus to be replicated, and then that causes more viruses to form. Eventually, the cell bursts. It lyses, okay? Bursts out, and that DNA that was all in that donor cell gets packaged up in those viral particles. Now, the virus injected the first cell with its DNA to make more viral particles. But when all that DNA got mixed up in the second stage, and then lysis and all those viral particles were formed, many times pieces of the donor cell's genetic information gets into the viruses too. See this one right here? Now then that goes in and attaches to another cell and injects not viral DNA, but actually DNA from the host cell back here. And now these letters are representing alleles, genotypes. That's why we call viruses vectors. They move things around. They can move genetic information from one cell to the other. It's kind of like the same term, well, it is the same term we use for insects that can transmit disease, like malaria, the vector. They move it. Okay, any questions on that? So we got transformation, conjugation, transduction, and of course just the whole thing. They reproduce really fast, so you get plenty of variation in these guys, and you're making lots of offspring. Let's talk about how they make a living when it comes to nutrition and metabolism. It seems like, oh man, just seems like the prokaryotes found every way to eke out a living. That's why I think I find them so interesting. This chart looks a little bit intimidating at the beginning, but it actually isn't quite that bad. It's got a lot of pictures on it to show you some examples and to give me talking points. But if you're trying to think about separating these different ways that nutrition and metabolism occur among living critters, take away the pictures, that's what we're looking at. But let's go back to the bigger chart. Organisms make a living on Earth through a combination of acquiring carbon. Carbon is necessary. All living things on this planet use carbon, or carbon-based life form. And we use those carbon atoms to make molecules, and we link those molecules together to make bigger molecules, and all that stuff you learned about during the monomer polymer stage of your life back there in Bio 1. And you know, the things you're going to need to be able to build things are, number one, materials, carbon, number two, energy. Now, you can get that energy from breaking down other molecules like we do, food, or you can get it from the sun. So let's take a look at the different ways organisms, not just prokaryotes, but organisms in general. Let's start with where you get your carbon, and let's assume you get your carbon from carbon dioxide. So if you're thinking plants, then you'd be right. If you get your carbon, your source of that very important essential element, from carbon dioxide, and you use energy to build up more complex molecules, you're what we call a photolite autotroph. See, in the little blue box in the middle, I've kind of put how you put the words together. So if you're a plant, for example, and some protozoans, but mostly plants that we're familiar with, you use light, photo, and you use carbon dioxide from the air, auto, you're a photoautotroph. So that's plants, some protozoans. This is seaweed, kelp. Kelp is actually a protozoan, and that's a photoautotroph. Other protozoans like Euglena, the little blue arrows here, you notice, there are some who actually have more than one way of making a living, so we'll talk about that in a second. Among the prokaryotes are cyanobacteria, the photosynthetic algae like Oscillatoria and Anabina. There are things we call purple photosynthetic bacteria, the Rhodobacter, and then the plants. All right, now, if you use carbon dioxide, but your energy comes from inorganic oxidation, in other words, breaking down inorganic molecules, then you're really kind of a special critter. For one thing, you're only a prokaryote. Only prokaryotes can engage in chemoautotroph. Okay? Chemoautotrophs, like methanogens, what they do is they hit carbon dioxide and hydrogen, they stick those together to make methane, and that releases energy. That's the smell sometimes when you're walking through a swamp. There's a lot of methanogens that live in a swamp. There are sulfur bacteria that do that. They actually take sulfur or hydrogen sulfide and they oxidize it. Remember, when you oxidize something, you release energy. And the guys down below, the NH3, the nitrifying bacteria, oh my God, for plants, those are absolutely essential. So we'll talk more about those when we get into the plants, but these are the guys that make nitrogen available for plant growth. You know, nitrogen is limiting. It's why we put fertilizer on our plants, because they need nitrogen. Okay? The other side of the equation is if you get your carbon from other organic compounds. So you're a hetero in terms of your carbon source. You don't get it from carbon dioxide. And if you use those organic compounds and the carbon in them to make up other molecules by oxidizing things, then you're a chemoheterotroph. Now, you're either a chemoheterotroph who oxidizes organic things, like us, animals. See the guy eating the burger? You're eating meat. You're eating something organic. Fungi. Some plants, like that white one there, that's a Indian pipe. Actually, some grow on campus, and they actually are heterotrophic plants. They're not photosynthetic. They're white, right? They can't photosynthesize. This is where the double dipping occurs. That little green thing, Euglena, is another one of these organic chemoheterotrophs. Paramecium, this is botulism. This is the prokaryote that causes botulism. You could also be an inorganic chemoheterotroph. So this time you don't get your energy from food. You don't get your energy from organic materials, carbon and hydrogen. You get them from oxidizing sulfur, or which you then use to oxidize other organic compounds. That's why this one is different than the purple sulfur bacteria. So you oxidize sulfur. The energy from that, you oxidize other organic compounds. And then my favorite of all, at the bottom, Halomonas titaniae, they oxidize iron. They oxidize iron off of the, they're only found on the Titanic. They oxidize the iron found in the metal plates of the Titanic. The energy from the oxidation of iron they use to oxidize other organic compounds found in the environment. Very cool. Another one that's only in the prokaryotes is an organism that is a photoheterotroph. So they get their carbon from some other organic compound, and this is another double dipper. This is the purple photosynthetic bacteria. I say double dipper because Rhodobacter can do it this way or this way. So the pictures and examples, again, are just my way for talking points. But if you want to break it down organizationally in your mind, you can simplify the chart to this. So it depends on where you get your energy and where you get your carbon, okay? Folks, I'm at time, so that's a good place to stop right there. I'll go ahead and finish the recording. You guys have a good weekend. We do not have the lab practical next week. We talked about that already in class, so don't get yourself all freaked out. We're going to move that a week later. Obviously, we haven't done lab 4, so we can't. So let me stop.